Welcome back to this next video, and in this video we are going to talk about the Campylobacteriosis, and it is a disease which is caused by the Campylobacter jejuni. Now, when you talk about the foodborne diseases, these foodborne diseases, they can be defined as the illness due to the ingestion of spoiled or poisonous food contaminated by microorganisms or toxicants, which may occur at any stage during the food processing from production to consumption. So these foodborne diseases, they can be because of the poisoning of the food, it can be because of the food infections. Now, when you talk about the food poisoning, these food poisoning can be the chemical poisoning, some kind of the toxicants they have been added to the food, which is causing the foodborne diseases. Or this food poisoning can be the biological poisoning, and when you talk about this biological poisoning, it is actually because of the poisonous animal tissue or the poisonous plant tissue. The second important reason of the foodborne diseases, they are the food infection. What I mean by that is when the food get contaminated by various pathogenic organisms, and when you ingest that particular food, that is going to cause the foodborne diseases, and that will be the focus of this particular video, how the pathogenic organism. In this particular video we'll be focusing on the Campylobacter jejuni, and in the following videos we'll be focusing on some other important microorganisms, the pathogenic microorganisms, which when contaminate the food can lead to the foodborne diseases. Now, which bacteria they are responsible for the foodborne illnesses? Now, when you talk about the types of the bacteria, there are thousands of types of bacteria which are naturally present in our environment. Now, not all of these bacteria they cause disease in humans. For example, some bacteria are used beneficially in making the cheese and yogurt, but those particular bacteria that have the potential to cause disease, they are known as the pathogens. And when these pathogens enter the food supply, they can cause the foodborne illnesses. Now, of these thousands of the types of bacteria, only a few types cause millions of cases of foodborne illness each year around the world. Ironically, most cases of the foodborne illnesses they can be prevented when you follow the proper cooking or the proper processing of the food, so you can destroy these bacteria which are responsible for causing the foodborne illnesses. Now, these bacteria, they can theoretically be in any food. They can grow in just about any kind of the food available to them, but they are very fond of protein foods. And when you talk about the foodborne illnesses, they are mostly because of those particular foods which are rich in protein, such as the meat, the poultry, the seafood, the eggs, and the dairy products in particular, as well as high protein vegetables such as the beans and the grains. So these bacteria which are responsible for causing the foodborne illnesses, they are mostly found in those particular food which are rich in proteins. Now, how these bacteria, these disease-causing bacteria, they get into our food. Now, bacteria may be present on products when you purchase them. For example, the raw meat and poultry during the slaughtering process, they may be contaminated with these pathogens. These bacteria may be present on the seafood and eggs, and they are also not sterile. Neither are products such as the lettuce, tomatoes, sprouts, and melons. What I mean by this is that when you purchase a particular food, you have to properly wash it, you have to properly cook it, because if these bacteria, if they are not properly treated, they can be responsible for causing the foodborne illnesses. Now, another way the bacteria they can enter into our food, they are from animals which can contaminate the food. That is when water used to grow fruits and vegetables is contaminated by animal feces. For example, if the animals they are having these foodborne illnesses, and if you are washing them and that particular water is used to grow the fruit and vegetables, the feces of those particular animals, they can actually contaminate the food, and they can be responsible for getting into the food, and hence causing the foodborne illnesses. Now, foods including safely cooked, ready to eat can become cross-contaminated with bacteria when introduced on the raw products, meat, juices, or other contaminated products, or by poor personal hygiene. So, if you summarize this whole story, these bacteria, they can be present on the raw meat in the poultry or the seafood. They can get into our food by the animal feces, or by contaminated products, or by poor personal hygiene. Now, these bacteria can contaminate food via the food storage facility as well. Now, one of the important terms that is used to classify or to identify the bacteria via the food storage, that is known as the danger zone. And this danger zone actually refers to the temperature in which these bacteria, they are stored, or the food in which the food which are contaminated by these bacteria, that is stored. Now, these bacteria, they multiply rapidly between 50 and 140 degrees Fahrenheit, that is between 10 and 60 degrees. To keep food out of this danger zone, the cold food, they should be kept cold, and the hot food should be kept hot. What I mean by that is that you should keep the food below 10 degrees centigrade or above the 60 degrees centigrade, because this is actually the temperature where bacteria, they rapidly multiply, and when they multiply, they can be responsible for causing the foodborne diseases. Now, you have to store the food in a refrigerator or freezer, that is 40 degree Fahrenheit or below, or in a freezer from 0 degree Fahrenheit or below. Now, cook food to about 160 degree Fahrenheit, and the temperature for the roast, steaks, and chops of beef and lamb, that is about 145 degree Fahrenheit. Now, maintain the hot cooked food at 140 degree Fahrenheit, and reheat the cooked food to 165 degree if that is present in the refrigerator. You want to reutilize that, you have to reheat the cooked food to about 165 degree Fahrenheit. So this is the danger temperature zone. So you have to keep the food in below these particular range, below the 10 degrees and above the 60 degrees centigrade. Now, if you talk about the most common foodborne pathogens, so according to the Center for Disease Control and Prevention, the CDC, the most common pathogens responsible for foodborne diseases, they are the norovirus. It is one of the important viruses responsible for causing the foodborne diseases. The Campylobacter jejuni. Among bacteria, the most common cause of foodborne diseases is the Campylobacter jejuni, which we'll be focusing on detail in this particular video. You've got the Salmonella, the Clostridium perfringens, the Staphylococcus aureus, Clostridium botulinum, and E. coli, especially the O157H7. So these are the most common foodborne pathogens according to the CDC. Now, when you talk about the Campylobacter jejuni, it is responsible for a condition which is known as the Campylobacteriosis. So, Campylobacteriosis is an infection by the member of the Campylobacter genus, most commonly by the Campylobacter jejuni. Now, this Campylobacter jejuni, it is amongst the most common bacterial infection of humans, or this Campylobacteriosis is among the most common bacterial infection of humans, often a foodborne illness. Now, these during the Campylobacteriosis, an inflammatory, sometimes bloody diarrhea or dysentery syndrome that is generated, mostly including the cramps, fever, and pain. Now, this C. jejuni, it is one of the most common causes of food poisoning in Europe and US. And the vast majority of the cases of C. jejuni that occur as isolated events are not as part of the recognized outbreaks. The European Food Safety Authority, they estimated in 2011 that there are approximately 9 million cases of human Campylobacteriosis per year in the European Union. So this is a very huge number of the cases, but these cases usually occur as isolated events and they are not recognized as outbreaks. Now, this C. jejuni, it is a commensal bacterium that resides in the intestinal tracts of many wild and agriculture-associated animals, and there it is not causing any kind of the disease, but it is living as a commensal bacterium. Now, the poultry flocks, especially the chicken, they are commonly colonized with this C. jejuni, but again, this colonization is commensal in nature and it is not creating any problem in these chickens. Now, as such, handling and consumption of poultry meats contaminated with C. jejuni are leading risk factors for sporadic cases of diarrheal diseases caused in humans. So they are present in the commensal form in the intestinal tracts of the chicken, but if you are not properly dealing them, that particular C. jejuni can enter into the poultry meat, and that is responsible for causing the diarrheal diseases in humans. Now, outbreak cases of diseases are often attributed to the consumption of contaminated water or raw milk. We will talk about the different sources from where the Campylobacter can enter into our system, enter into our body through different sources. We will discuss that. Now, the rates of diarrheal diseases caused by C. jejuni, they are high in both developed and developing countries. So the infections of the C. jejuni, they are there both in the developed and developing countries. When we talk about the biochemical properties or the laboratory identification of the Campylobacter jejuni, so this Campylobacter, it means the curved rods. And this Campylobacter term is derived from the Greek, the campylos, which refers to curved structure, and bactron, which is actually referring to a rod structure. Now, there is a wide diversity in the genus of Campylobacter, and the species are metabolically and genetically different to the extent that one can question whether one genus is adequate to house all of these species, because they are metabolically and genetically very different from each other. Now, C. jejuni is one of the most important from both a microbiological and public health perspective. As I've told you, it is responsible for millions of cases around the world per year. Now, this Campylobacter, this is the Campylobacter jejuni, is a helical-shaped bacteria. It is a non-spore-forming bacteria, and it is gram-negative in nature. So when you are going for the gram staining of the Campylobacter, you'll be seeing these kind of the rods under the microscope, and the shape of the bacteria, that will be helical shaped over here. Now, the Campylobacter jejuni is a microaerophilic bacteria. What I mean by the microaerophilic is a microorganism that require oxygen to survive, but require environment containing lower level of oxygen than that are present in the atmosphere. So when you talk about the atmosphere, there is about 21% oxygen in the air of the atmosphere, but these microaerophilic organisms, they typically require between 2 to 10% oxygen. So when you grow them in a medium, so as you can see over here, that will not grow at the top of the tube, because if they are growing at the top of the tube, the oxygen that will be required to them, that will be 21%. So and they are growing in this particular area away from the top, that means they are avoiding the oxygen to up to the 21% level, and here the level of the oxygen, they are low, so they are very good at growing at the microaerophilic environment. Now, these Campylobacter jejuni, they are non-fermenting bacteria forming motile rods, as I've shown you in this particular image, that they are forming these rods, and they have amphitrichous flagella. The amphitrichous flagella mean that the Campylobacter jejuni have one flagellum coming from each end of the cell. So if this is one end of the cell, there is one flagellum. This is the other end of the bacteria, there is another flagellum. So this particular phenomena, or this particular condition of the flagella, that is known as the amphitrichous flagella, that there is one flagellum coming from each end of the cell. So Campylobacter jejuni, they have these amphitrichous flagella. The Campylobacter jejuni is oxidase positive. What I mean by oxidase positive is that it produces the cytochrome C oxidase enzyme. And what this cytochrome C oxidase enzyme do is that if this is a paper that has been encoded, or that is having the tetramethyl phenyl p-phenylene diamine, or this dimethyl p-phenylene diamine, if that particular chemical is present in this particular piece of paper, and if you apply the organism and that organism is oxidase positive, what I mean by that is if that organism is producing the cytochrome C oxidase, the cytochrome C oxidase will act on this particular chemical, thereby converting it into a colored product. If the organism is negative, the cytochrome C is absent, that means there is nothing to interact with this particular chemical, and if there is nothing to interact, it remains colorless. So the oxidase positive organism give you the colored product, and the oxidase negative organism give you a colorless, that is colorless because there is no enzyme to act on this particular dye. Now the Campylobacter jejuni, they grow optimally at 37 degrees centigrade to 42 degrees centigrade. So this is the optimum temperature requirement for the growth of Campylobacter jejuni. Now there is a selective media which is known as the Campylobacter selective agar, that is used for the selective isolation of C. jejuni from the fecal or rectal swab, and this media inhibit the growth of the other bacteria. So this selective media can be used to isolate the Campylobacter jejuni from the other bacteria, because this media is going to allow the growth of only C. jejuni and not any other bacteria. Now the virulence factor of the Campylobacter jejuni, there are a variety of the virulence factors. Now the first and the important virulence factors, they are known as the adherence and the invasion virulence factors, and these virulence factors, they are responsible for adherence to and invasion of the human intestinal epithelial cells. Now the C. jejuni that possess at least two outer membrane embedded fibronectin binding protein, which is known as the CADF. This is the first outer membrane protein, which is known as the CADF, and the CADF is for the Campylobacter adhesion to fibronectin. And another important outer membrane protein, that is known as the FLPA, which is for the fibronectin-like protein A. Now both of these proteins, this CADF and this FLPA, that are present on the outer membrane of the bacteria, outer membrane of the Campylobacter jejuni, and they have the ability to bind to fibronectin. This is not fibronectin, this is fibronectin. You should exclude this O. So both of these, the CADF and the FLPA, they have the ability to bind to the fibronectin protein. Now this CADF gene, it is about 960 base pairs, and the protein that it encodes is having a molecular mass of 37 kilodalton. So CADF is a 37 kilodalton protein. The FLPA gene, it is 1236 base pairs, and the molecular weight of the FLPA gene, FLPA protein is 46 kilodaltons. Now this CADF, it mediates binding to fibronectin utilizing a 4 amino acid motif. There is a 4 amino acid motif in the CADF through which it binds to the fibronectin, and this 4 amino acid motif is phenylalanine, the arginine, the leucine, and the serine. So the CADF is utilizing this particular 4 amino acid motif to bind to the fibronectin. Whereas the fibronectin binding site within the FLPA, it has been localized to a span of 9 amino acids. So here you have got big motif as compared to the CADF, because this is made up of 9 amino acids, and the 9 amino acid motif is the this one, the TRP, the ARG, the protein, the histidine protein, aspartic acid, the phenylalanine, the arginine, and the valine. So this is the 9 amino acid motif which the FLPA utilize to bind to the fibronectin binding protein. Now how the CADF and the FLPA, they are actually helping in the adherence and invasion of the human intestinal cells. Now, I have a detailed discussion on this in my last video, and I'll share the link in the description, that when a protein that has the ability to bind to the fibronectin, they actually can remodel the cytoskeleton, especially the actin filaments. And when a protein that has the ability to remodel the cytoskeleton of particular human cells, particular intestinal cells, those remodeling in the cytoskeleton is actually allowing the bacteria to enter into the cell. So when the CADF and the FLPA gene, they bind the fibronectin, that particular fibronectin is going to bind to the integrin receptor present on the human cells, present on the human epithelial cell in this particular case. And when the fibronectin bind to the integrin receptor, that is going to cause the remodeling of the cytoskeleton, and that remodeling is actually allowing the bacteria to enter into the cell. Now, how are we sure that the CADF and the FLPA gene, they are responsible for adhesion and invasion into the human cell? Is because when you disrupt either the CADF or FLPA, when you mutate them, that actually results in the C. jejuni mutants. They are impaired in their ability to bind to cultured epithelial cells. So in the absence of the CADF and the FLPA, the mutant C. jejuni, they do not bind to the epithelial cell. That means if they do not bind, they cannot remodel the cytoskeleton, and hence they cannot enter into the epithelial cells. We will discuss about the other virulence factor in the next video. And if you like the video, please subscribe to my channel, hit the like button and share it with your friends.